Well, hello, everyone. I'm glad to see that there are some people still in the room. And I'd like to thank my former boss, actually, for giving me such a good introduction to my talk, because most of what he talked about, I'm actually going to recapitulate. So if you didn't get it the first time, you will get it the second time, hopefully. So the title that you see in your booklet is a little bit of a misnomer, because I will give a regulatory overview. I will give a little bit of a glimpse of what the office is currently working on with regard to TTC, but I'm not going to give you a peek into our future plans for guidance formulation. Sorry. That would not be allowed. So with no further ado, let me continue. So here's what I'm going to talk about. I'm going to give you a brief introduction as to how we do things. Then I'm going to talk about the safety assessment process for directs and then for food contacts. I'm going to talk about the ongoing projects and some final thoughts. So briefly, we do both pre-market and post-market evaluation in OFAS, but our primary concern is pre-market evaluation. And we evaluate all things, all food ingredients, according to the legal safety standard of reasonable certainty of no harm under the intended conditions of use. And as part of that evaluation, we look at the structure of the compound, its physicochemical properties, that includes particle size, as appropriate, dietary exposure, and the available toxicity data for compounds and structural analogs. And then we look at both neoplastic and non-neoplastic endpoints. We consider carcinogenicity to be the most sensitive endpoint and the most concerning endpoint. We do not use a threshold in our evaluation of carcinogenicity most of the time. And we do use genotoxicity assays, in vitro assays, and in vivo assays as hazard identification for that endpoint. And we also consider non-neoplastic endpoints, such as developmental toxicity, reproductive toxicity, and neurotoxicity as appropriate. Allergenicity if it's a protein. Oops, wrong way. And so here's where OFAS safety paradigm and the TTC approach, I'm going to talk about the similarities versus the differences. As Mitch pointed out, the TOR approach actually originated with the FDA, but it is considered a risk management approach. Just because something the exposure is less than 0.5 parts per billion doesn't mean we don't evaluate the safety at all. We do look at toxicity data for all exposures, but we simply, the TOR is for most chemicals is meant to say, okay, we don't need you to submit toxicity data for this chemical if the exposure is below this particular level. That doesn't apply. For some chemicals, that does not apply. So again, it's a risk management tool. As far as the similarities go, there is not an exact correspondence to the TTC approach as explicated by the Monroe and Crow's working group. But there are similarities and differences. So similarities is the science-based TOR approach, which is used as a risk management tool. We assess concern levels for given exposure based on structure and the available toxicity data. That's very similar. And we use a structural classification scheme that's based on Kramer et al., the 1978 paper. Differences. We use defined exposure tiers as thresholds for the need that trigger the need for toxicity testing. So it doesn't mean that we say, oh, we're not evaluating any data for this endpoint. These are thresholds for saying we need you to provide us a study for X. And then the second one is threshold values of structural class values differ from those as stated in Crow's et al. And frankly, some of the concern level values and the tox tier values some of those, I have no idea where they came from. It's lost in the history of time where they came from. They're not based on an explicit analysis like the TOR level was, or like the Crow's et al. Levels are. And then, and that's what I just said. So keeping that in mind, we will move on to, God, keep doing this, to the safety assessment process for food additives, and you're going to see something familiar. If you're awake during Mitch's presentation, you should recognize this graph. And this is a graph that is present in Red Book, and it is used in the evaluation of direct food additives and also indirects. I mean, just basically you can use it for anything. And it is these concern levels are used to describe the minimum set of toxicity tests that are needed to support the safety of a given exposure to a food ingredient. It covers a very wide CEDI range, and notice that I'm giving the CEDI range in terms of micrograms per kilogram body weight per day. That's not an accident. We used to be giving them in micrograms per person per day. However, we are starting to move towards doing separate assessments for infants and children, and for the ease of comparison between exposure to infants and adults, we are now putting them in terms of micrograms per kilogram body weight per day, because the food consumption amounts per day for infants is much different than adults. So it's easier comparing apples to oranges if you use that metric. And note, this particular graph does not specify a level below which no tox data are needed. Let's see if I can do this. Okay, so here are the recommended toxicological testing. Here's the set of recommended toxicological tests. Note that when you have a low concern level, meaning usually either structurally non-concerning plus low dietary exposure, you only need to do genetic toxicity tests plus short-term toxicity tests. So you don't have to go through the whole battery of reproductive toxicity testing and neurotox and metabolism PK. It's very low. It's a very sparse set of toxicity tests. Whereas once you get into the high concern level, if you've got a major screaming structural alert, or if you have a really high exposure, you get the whole banana, up to and include the human studies. So that can get really expensive. So then now we move on to food contact materials. The difference between food contact materials and directs is that the exposures to food contact materials, for one thing, they're not deliberately put into food. Indirects are present in food as incidental. They migrate from the food packaging into food, whereas directs are put deliberately into food. So the exposures are much, much lower. The second is that usually for direct food additives, you're talking about structural types that are of very low concern. Sugars, artificial sweeteners, that type of thing. Whereas for food contact materials, you may be talking about some very, very, very toxic substances. So it's a somewhat different paradigm. So here's the way we evaluate food contact substances. We go from less than 0.5 to 1 part per million dietary exposure. Supposedly above 1 part per million, we're supposed to ask for a food additive petition, because that's getting into direct food additive territory. That doesn't always occur, but according to Hoyle, it should. So and then we assess we assess the need for data according to what are called tiered exposure ranges. So that EDI of 0.025, that corresponds to TOR. So anything below TOR, we assess just carcinogenicity. We don't recommend testing, but we still assess the available data. And if it looks like there needs to be some data to support safety, we still will ask below that level, depending upon what the data for structural analogs and the structure of the compound say. Then above TOR, we start talking about cumulative EDI, because when a lot of these compounds are exposure from multiple uses, multiple different FCSs, so it only makes sense to start looking at how much you're getting, not just from the proposed use but of that particular FCS, but from everything. So if you've got an exposure that's between TOR and 50 parts per billion, we start asking for gene tox, which is, again, hazard ID for carcinogenicity. And then once you get above 2.5 mics per kg per day, we start asking for systemic toxicity. And again, because we are fairly low exposure, we ask for subchronic data. We're not asking for carcinogenicity. We're not usually asking for developmental repro. But we can still ask for these assays if it looks from the available data plus the structure of the compound that we need them. So again, this isn't a rigid structure. It's a generalized framework upon which we fit the circumstances. So that's the food contact substances. And again, I can't emphasize enough, we really use structure activity relationship analysis to fill in the gaps, because a lot of times for what comes in, we may have data that we may have an Ames, but we don't have a gene tox assay in mammalian cells. We have something, a mammalian cell gene tox assay for a structurally similar compound. So we use structure activity relationship analysis both to fill in the data gaps and to identify the need for further testing if as appropriate. And here's some of the resources that we use to look for structural analogs and to identify data for those structural analogs that might be useful. And again, as I said, there are exceptions to that tiered toxicity testing paradigm. Biocides are intended to be biologically active. They're intended to kill cells. So because of that, we assume that the need for testing is 1/5 of the levels that would be identified for general compounds. Another one, long-chain perfluorinated compounds. They're biopersistent. So you can't use the same toxicity testing cutoffs for long-chain perfluorinated compounds as you would for other compounds, because they tend to accumulate in the body, and the daily dose does not correspond to the systemic dose that the person's actually feeling. So, and then the second one is structural alerts for neurotoxicity, organophosphates, and carbamate analogs. And then, of course, engineered nanomaterials, because we just know so little about them. So here we get to ongoing projects. There's Tim Adams, who is a contractor with our office, is trying to update the Kramer classification scheme. So we have an internal database called SERIES, which incorporates data from over 2,000 compounds and studies available after 1978, and incorporates data from submissions that we've evaluated, as well as data from other sources, so it's very valuable. And he's been analyzing all that data, and the results so far is that he is considering changing the number of chemical classes from 3 to 4 to better characterize the different toxicity threshold values, and updating questions in the classification scheme, and and then incorporating ADME data into the decision tree, which right now our toxicity tier guidelines don't really incorporate ADME data. That would change if this scheme is adopted, and it's being considered for use with all traditional toxicity endpoints. Again, this is very, very, very experimental work. This is not on the horizon anytime soon, so don't get all excited. We'll see whether or not it comes to pass, and it may come to absolutely nothing. Who knows? So that's one project. And the second project was the refinement of the cancer TTC dataset, and this was presented as a poster in SOT in 2012. And excuse me. The purpose of this analysis was to expand the cancer TTC analysis as looked at in Cheeseman et al., using bioassays from SERIES and the CPDB databases that met regulatory criteria, as well as incorporating data from Ames assays. So for the CPDB studies, they used the lowest statistically significant TD50 that were Red Book compliant oral toxicity studies. They did not include TBA, MXA, MXB, or equivocal studies. For the Ames data, they used data from series, and that met the criteria for study design. In other words, they would be considered Red Book compliant studies. And then they looked at how these assays were called. So if an assay was called positive by the FDA, then even if the same assay were called negative by the NTP, then FDA regulatory call would supersede all others. Then next in the hierarchy would be NTP, Ames studies, and then Tokyo ICANN and ISSCAN, and then the CPDB calls. So then they grouped the TD50 values by structural domain and compound source, and whether or not they had Ashby tenant alerts, which are structural alerts that are associated with carcinogenicity. And then they also grouped them by positive or negative Ames calls. And then they did a statistical analysis and derived the mean TD50 values for the following positive categories. So as you can see, the lowest TD50 value would be, as you would expect, those compounds with positive Ashby tenants and positive Ames. So these are directly mutagenic compounds. And that one had a TD50 of 100 mg per kg per day. And as you would again expect, if you don't have any Ashby tenant alerts or Ames alerts, you get a relatively high TD50 value. Now, surprisingly, if you have an Ashby tenant alert and negative Ames, you have approximately the same TD50. So it appears that the Ames from this particular analysis, the Ames call is more predictive than the Ashby tenant analysis. And then the final thoughts. As Mitch has pointed out, OFAS has been using a TTC-like analysis, a threshold-based analysis, all along. However, we are on efforts to integrate a more formalized, updated TTC approach into our safety assessment are currently ongoing. And with that, I will take questions.